Given lichens' ability to appear among the first colonizers of such inhospitable habitats, it has been suggested that they may have been pioneers of the move from the oceans onto dry land. They could have played an important role in weathering minerals and thus preparing the soil for vascular plants to grow on. Recent molecular phylogenetic analyses of the fungi involved and their photosynthetic partners suggest, however, that the evolutionary history of lichens is shorter than anticipated, but also more complex. Both fungi and photosynthetic microbes have been around for more than 1 billion years. The ancestors of today's cyanobacteria began two-step photosynthesis around 2 billion years ago, setting off the biological production of oxygen. Early eukaryotes engulfed these photosynthetic bacteria and evolved to become algae and plants. The first fungi may have split from the lineage that eventually led to animals at least 1 billion years ago. But when did fungi team up with either cyanobacteria or algae, as they do today with both, to form a symbiotic relationship? Dating the origins of lichens is important for our general understanding of the evolution of life on Earth, as lichens could conceivably have prepared the ground for vascular plants to expand on the continents around 440 million years ago. With their roots, vascular plants could substantially modify the land surface and make it habitable for other species. The key question, however, is, did lichens make the land habitable for plants? Scientists since the times of Darwin have thought, based on the pioneering role that lichen take today in inhospitable environments like Antarctica, that they may have been the first complex life on land. However, the fossil evidence to back up such speculation hasn't been found yet. Identifying early lichens from the fossil record is extremely difficult, and uncontroversial examples of early lichens are rare. A suggested lichen-like assemblage of filaments with algae that was described in 2005 and dated to 600 million years ago is not generally accepted as evidence of lichen. Subsequent molecular studies have begun to hint at younger ages of individual groups of lichen fungi. However, interpreting this information is difficult because several distantly related groups of fungi form lichens, raising the questions: how many times did lichenization evolve, and how old were these transitions? In a bid to clarify when lichens originated and vegetation first arrived on land, Matthew Nelson at the Field Museum in Chicago and colleagues calculated age estimates for phylogenetic trees, including most groups of present-day lichen-forming fungi and algae. Because of the lack of conclusive fossil evidence, the researchers used an alternate approach in which they placed molecular phylogenies of fungi and algae in a temporal framework by inferring the evolutionary relationships of extant taxa and linking them with fossil data. Nelson and his colleagues then inferred where and when in the fungal phylogeny lichenization evolved by using a process known as ancestral state reconstruction. The ages of nodes reconstructed as lichenized could then be placed in the context of land plant and terrestrial ecosystem evolution.